tardes a todos y disculpas por estos minutos de retraso. Quisiera introducirles, y tengo el gran placer de introducirles a ustedes, a Martine Hossaert. Haré esta introducción en español. Tardes a
Martine,
todos.
I will do the introduction in Spanish
because I assume everyone listening is Spanish speaking, and then you will go on in English. La presentación se hará en inglés. Martine Hossaert es directora y científica en el Centro Funcional de Ecología Funcional y Evolucionaria en Montpellier. Es también asesor científica del Instituto Nacional de Ecología y Ambiente del CNRS, que es el Centro, es el equivalente de CONICET en Francia, y es también responsable de dar lineamientos estratégicos y planes y programas en biodiversidad y de relaciones internacionales en el INE, que es este Instituto Nacional de Ecología y Ambiente. Martine es una especialista de ecología química, que es el estudio del rol de los señales químicos en la naturaleza y en la ecología, que tienen que ver con la defensa de los organismos y la comunicación e interacción de los mecanismos y organismos. Y para hacer eso ha dirigido un grupo multidisciplinario en Montpellier, en un centro que es un centro de ecología, es un gran centro de ecología en Montpellier. Específicamente ha trabajado en polinadores de higos, que son las avispas que polinan los higos, y ha estudiado esto en varias partes del mundo, como Taiwán, Nueva Guinea, Madagascar, Sudáfrica, África Central. Y también se ha preguntado acá sobre el impacto de la polución, por ejemplo, la ozono, en la comunicación de la polinización de los higos por estas avispas, que se hace mediante productos volátiles. Aparte de ser asesora científica en el CNRS, también define las estrategias de investigación en biodiversidad y propone nuevas iniciativas de ciencia de punta en lo que concierne biodiversidad, cambios globales, la salud ecológica y las soluciones basadas en la naturaleza. También ha sido responsable por los comités de reclutamiento en el CNRS y también ha participado en la distribución de fondos de las grandes agencias francesas y europeas, y ha representado Francia en varias ocasiones en las diferentes COPs. Para los del IMIBio, quizás se hayan encontrado en la COP14 en Sharm el Sheikh, y también ha estado implicada en el IPPBS 6 y 7, y ahora ha representado a Francia en Glasgow y va a representar Francia en Kunming, en China, el año próximo. So, Martine, the word is all yours, and you're very welcome, and we will listen to you with much attention, because this is a very interesting topic, and me as an immunologist working on lymphokines and cell-to-cell communication, I'm particularly interested in hearing what you are going to tell us.
So, yeah, Many thanks, Eric. And so I'm happy to show what I know and what I can share with you about the chemical mediation, and particularly the role of this chemical mediation in interspecific interaction, and with a focus on tropical ecosystem. Oops. I cannot move my slide.
You cannot move your slide?
No. Oh, yeah, it's okay. So when you work on chemical mediation, you work on a chemical ecology, and chemical ecology is to bring chemistry into ecology. So as Eric was saying, it's a totally interdisciplinary science, and bringing chemists with ecologists is sometimes a challenge. But we find very interesting colleague in chemistry that really want to work with us, and it was a very successful study right now in France. So chemical ecology is a study of how organic molecules can mediate the interaction among living organisms. And so chemical ecology is also at the heart of fundamental and new questions in evolutionary biology and functional ecology, what I'll try to show you today. So they have a very important role or place in the functioning of ecosystem, and also in the origin and the maintenance of the biodiversity. So chemical ecology, started not too long ago if you compare to other, science. The first step in first discover in chemical ecology was in 1959, where Adolphe Butenandt discovered a new molecule that he called bombicol, that was very important as a sexual attractant for male of this butterfly, Bombyx mori. And it was the first study who showed how this molecule was mandatory for this attraction. But you have to wait a little before people really understand and really start to work on that. And it's only in 1983 that was founding the International Society for Chemical Ecology. And this society in 1989 have this declaration where they say that natural products are important, and they constitute a treasury of immense value also for to humankind. So at the beginning, chemical ecology was more to describe many natural substances and how they can be used for human. And of course, you have the famous example of the acid salicylic that people coming from salix, and that people know for a long time and use it. But some people were discovered this molecule of salicylic acid, and of course it's used for this very common and everyday medicine called aspirin. And little later, you have also, this team was a French team that using Taxus baccata discovers this more sophisticated molecule called taxotere. And during 20 years, it was a more important molecule in cancer, not prevention, but cancer cure. So, and the CNRS get a lot of money from this molecule, but it's no more now. We have new medicine for cancer, but it was very important for a long time. But chemical ecology is not only to do this inventory of natural substances, was quite important, but there is also other aspects that, as a chemical ecologist, I am also very interesting. Chemical ecology is at the heart of innovative and fundamental questions, as I said before, in evolutionary biology, and I am an evolutionary biologist, and in functional ecology. And so what I want to do is to show you some example using, things that I'm working on for a long time now. It's plant and insect interaction, just such as pollination or multitrophic interaction. So in plant and insect interaction, it's a use of what we call secondary metabolites, which are not really secondary, but we have. So people now are just calling it metabolite, but secondary metabolite is what you find mainly in the literature. And these metabolites are very important in interspecific interaction. Today I will speak only about interspecific interaction, and you have to know that this metabolite can be also very important for ultra-specific interaction, but I cannot cover everything today. So, and this secondary metabolite are used by plants for protection and also for reproduction. For protection, these metabolites are used for pathogen resistance. They are used also and very well known, and there is many studies on that, on herbivorous protection, like production of tannins or even more dangerous or more toxic substance. And but they are also used as signal for reproduction and used in pollinator attraction and also seed dispersal, attracting the insect or birds or bats that will disperse the seeds. And today I will also focus mainly on reproductive interaction. And so you have many insects that visit flowers, and they visit flowers, to flourish, to look for resources, and they can eat the flowers. They are looking for nectars, but also, they are used for reproduction. And you have all these very nice and intriguing stories on, deception, with the signals that attract, for example, fly with the smell of dead meat. And you have also all these obligate interactions that I am more specialist on it. So today you will listen a lot about this obligate mutualistic interaction. So in this mediation, floral display are very important. And so people know a lot about colors, but very little when I start, very little was known. It's not the case now since 10 years, but very little was known about odors. And I don't know why, because odors is an unlimited universe of odor blends. You have more than now 2,000 compounds that are described in floral odors. And of course, with so many compounds, you can make huge combinatorial potential. And they can be used also for long distance detection, longer than visual, for example, signals. So all these characters make a good template for fine-tuned recognition in specific pollination. And they are also used they are also very important for reproductive isolation on plants. And they can, for example, flowers are producing very specific blends that are a barrier for and isolate the flower from interspecific pollination, and also they are a filter to find the good pollinator. So when you are a flower and you want the right pollinator to come, you have to find this right partner. And this partner have to find you in the olfactory landscape. You can facility imagine that in tropical forests you have many plants that are producing this chemical signal. So in order to attract the right partner, you have to find this very fine tune. And also you want to have some constancy, because you don't want to have too much of spatial variation of your floral phenotype if you want the right pollinator to come. And also you want to have a dynamic emission and synchronize the the odor emission with the pollinator activity, and sometimes, use it as a repellent. So from the plant point of view, you have a lot of biosynthetic possibility, and that make combinatory possibility of traits that can increase your reproductive success. You can fine-tune more and more use signal for pollinator. But in our team, you also look at the both sides, for the plants, but we also look at the insect point of view. And so the insect has to learn how to do this floral choice. And you have innate preference, but you have also olfactory learning, like you can imagine for bees. So there is many selective pressure on both partners to favor a very clear signal and a mutually adaptive coevolution of floral signal and the perception by pollinator. And this is particularly the case for nursery pollination mutualism, where both partners depend of the other one for reproduction and survival. So my magic and wonderful system, as Eric was explaining, I work mainly on fig and fig wasp. So I don't know if you know all the story about this pollination mutualism of fig and fig wasp, but figs depends on a minute insect, 1 to 4 millimeter, and that these insects are bearing pollen. And it's the only insects that I will enter, because I will explain to you, but the fig are a close inflorescence, and it's the only insect that will enter the fig to pollinate. And of course, as usual in biology, you have some exception, but the rule is 1 species of pollinating wasp for each species of figs. And you have like about 800 species of figs, so you mean 800 species of pollinator associated to these figs. And the fig wasp only reproduce within the fig. So both partners need the other one for reproduction. So as I was just saying to you, figs are not only fruits. Many people know figs are fruit, but they are also inflorescence. So they are close inflorescence. So here you have, you have the bract, and that this bract close the inflorescence, and now it's called an ostiole. And you have here you have the inters, so the male function of on this inflorescence, and here all these little pink dots are flowers, and they are tiny flowers with an ovule and a style. And so, as I said just before, the pollinator will enter through this ostiole, and it will pollinate some flowers, and it will also lay eggs in other flowers. So both are involved in reproduction. We not have time today to explain all the cycle, but at the end, when the fig becomes a fruit, you will have seed within the figs from the flowers that have been pollinated, and where the insect lay eggs, you have a new generation of fig wasp that will exit, and will exit the fig in search of another fig tree. So, and fig tree, they are also very diverse. You can have this big, queen of the forest, this huge tree. I miss one. But you have also this hemiepiphyte that we call also strangler figs. So everything starts with a little seedlings that germinate, in a cracks. And after that, the seedlings send all his root around the tree, and the tree at the end will die. And also you have this hemiepiphyte of figs that can also, get around, some, building, or like here around some statue like in Thailand. This place is very famous for its temple. But also you can have tiny fig tree like this one in a river, and it's a very little, 50 centimeter shrub. You have also figs that are on the trunk that we call cauliflower figs. So you have also figs that grow on the ground, almost like strawberry. And you can also weedy epiphytes that climb for example, here, of course, on building, and that are used in Miami as an anti-graffiti tree. So, you know, figs are very diverse, and it's the reason why they are so fascinated. You know, so many species, so many very tight insect pollination, and a very diverse form of living forms. So makes this system very interesting, and that's the reason why we study it. And also, of course, because we have this very specific interaction between an insect and the pollinator that we'll find in the fig is braining site, and of course he will transport the pollen of the fig to another fig in an obligatory and specific interaction. And just remember you that you have 800 copies of this co-evolved mutualism. And all these reasons make it a very interesting model for evolutionary biology, but also for chemical ecology, because you have these 800 species of insect who have to find, you know, mainly in rainforest, only their own host and enter, you know, and you can guess that when a wasp enter in fig, it will stay inside. It will be very difficult to get out. Just to convince you, I have a very short movie on a fig wasp, so trying to enter a fig. So here it's a fig, and the fig wasp is entering through the ostiole, and you can see it's a very difficult task. And doing that, the fig wasp will lose his wings, and will lose his antennae, and so very often after having pollinating the flowers and laying its eggs, the fig wasp will die within the fig. So for all these reasons, we were seeking, it's a very interesting system to study all the chemicals that allow this interaction. And as you can see on this picture, you know, figs are very often green, you know, things in a green rainforest. So that's the reason why we were seeking that, you know, color is not very important to attract pollinator, but of course, odor will be the cue for this attraction. So it's a way we are doing it in the rainforest. We go with our little equipment, so we enclose, so that we have cauliflower fig in that case, and we enclose the fig in very special inert plastic bags. And in the bag, you know, we push air and we collect the air at the end of it. And you cannot see here, but you can see a little. You have a very special filter who absorb all the volatile compound emitted by this receptive fig, exactly when they are at the good stage to be pollinated. And so after that, we put all the filter in our bags and come back to Montpellier, and we do a gas chromatography and a mass spectrometry analysis to know exactly, you know, what are the compounds that the fig here were emitting, and we can describe what we call the odor bouquet. So we do that for many species, and of course we start, there is only one Mediterranean ficus species that we have in our experimental garden. It's Ficus carica. It's the one that you even can eat a fig if you want it, if you like it. And so we start with this species, and we find doing exactly what I was saying as before, and we find that this Ficus carica emit, you know, several compounds, but we were quite surprised at the beginning because, you know, these compounds like linalol or beta-ocimene or benzyl alcohol are quite common compounds. So at the beginning, because their interaction is so tight, that we are expecting, and so specific that we are expecting, you know, very special molecule, but that was not the case. It was a mixture of several common volatile compounds that we call VOCs. And so we went to mainly in Asia, where it's where figs species are originated, where you have the more diversities with several places in India, in Thailand, in Taiwan, in South China, in India, and so on. And we collect many blends of fig species. So here you have, for example, the example of 7 fig species collected mainly in Thailand and Yunnan in China. And you can, so that what I am showing to you, it's a principal component analysis with other blends that we have found. And you can really see with the different colors that each ficus species has its own odors. So, for example, this one, of course, Ficus auriculata, is very different from this other species, this Ficus exasperata, and from also this species, Ficus semicolata. And these 2 species, for example, the red one and the pinkish violet one, are in the same place, so we collect them in Saint-Patrick in the same garden. So the specificity of the fig and of its pollinating wasp is tuned by this chemical mediation, because each fig species has its own signal. And we did more and more analysis. Now we have like the volatile compounds of almost 50 fig species, and each fig species has its own odor blend. And so we can say that the chemical mediation facilitates partner encounter, and the chemical signal as a key factor maintaining the specificity of the interaction. And there is a selection for a strong and simple signal, but a quite huge quantity. And the simple signal is also an unambiguous signal in a very complex environment that you can find in tropical rainforest. But there is also some case of what we call private channel. And for example, that's the case of species. I was the violet species I was showing to you. So this species was quite unusual for figs, and produce a very rare in terms of figs compound. It's a 4-methyl anisole. And as you can see here, it's producing, almost, all the blend is made of this 4-methyl anisole, like more than 90% of the blend is made of this compound. So you have, mixture of simple compounds, or you can have also very specific compounds. So, but I told you that we work on both all the time. We work on the plant, but also we work on the insect, how the insect react to this odor blend. So I don't know if you know enough about physiology or odor reception of insect, but insects have a very specific olfactory receptor that send message to odor binding proteins, and of course go to the brain of these insects. And with this projection, they can recognize or not, some chemical compounds, and even mixture of chemical compounds. So it's what I will try to show you next. So we have this quite sophisticated equipment in our lab in Montpellier. Of course, we are not able to do that everywhere because you need to have all the equipment. But so here you have a fig wasp. And we have a very good technician that is able to put electrodes inside of the antennae of this. I remember it's 1 to 4 millimeter long insect. So you can imagine, the length of this antennae. So we put 1 electrode in the antennae, and during the same times, we send also odors of the fig on this antennae. And so we have a gas chromatography here, and during the same times you can register the chemical compounds that we find in these figs. And on the other side, you can also have the answer of the response of the antennae of the fig wasp. So so for all the compound that a fig can produce, you can say, okay, it's this one and this one and this one that the insect can recognize. So if you want to do some behavioral test with a fig wasp, we don't need to use, of course, the ones that the antennae are not able to recognize. So we can say that using this equipment, for example, for Ficus carica and its pollinator, Blastophaga senes, you can see that this pollinator only detects for 5 VOCs, 5 of these volatile compounds emitted by receptive pigs. And already told you these VOCs are quite common in plants. So there are the benzyl alcohol and linalool and the oxide of linalool. So what you are able to do after that is to have a less sophisticated equipment, is to do like in a wide tube olfactometers, is to do choice taste using a control and using here a dispenser of odors. So, and we can test, where the fig wasp will want to go, to the control of or to the odor, using each box, the linalool, the benzyl alcohol, the oxide, or the blend of the different VOCs that we make in a similar proportion of the fig we analyzed before. So this is our result. You can see that the blind, B1, so it's an odor choice. So you have 2 arms on the olfactometer. So you have the number of fig wasps that will go, the percentage of the fig wasps that will go on the arm with the odor, or here the percentage of the fig wasp will go in the control, in the blank with no odor. So for example, you can see that the blend in B1 is quite attractive for the pollinator. And if you put only one compound, the linalool, it's no more attractive. But the benzyl alcohol is quite attractive for pollinator. And the linalool oxide are not attractive. And if you compare the attraction between the benzyl alcohol and the B1, so our blend of compounds, you can see that the blend of compound is more attractive than the benzyl alcohol itself, only one compound. And if you compare even our blend to the real odor of a fig, we put a fig in the vials instead of chemical compounds, and you can see here that our blend is as attractive, even a little more, but not significantly. Our blend is as attractive as figs. So within this result, you can now know how the insect will react to this chemical signal, and demonstrates that pollinator is only attracted by a blend of 4 of these volatile compounds among, the 50 volatile compounds that the fig are emitted. So we also, play a little around this blend, and we change, the composition of the blend so that we have the original blend. And we make another blend where we change the benzyl alcohol proportion, and this blend is no more attractive for the pollinator. So we play around, changing the concentration of benzyl alcohol, changing the concentration of the oxide of linalool, and somewhere attractive, but it's when we increase a little of linalool. But it's really this ratio of the 3 VOCs, benzyl alcohol, linalool, and linalool oxide, that is determinant for the pollinator attraction. So going from a large bouquet of almost 50 compounds, we are able to show that only the ratio, and we need to be very specific of this ratio of these 3 compounds, is important for pollinator attraction. So that's what happens in the rainforest, and as you know, you are now confronted to a lot of ozone, a lot of pollution in our ecosystem, and so among this all these pollutants, the one who is quite important, at least for chemical compounds or volatile compounds, it's this ozone, because, you know, it's an O3. So if you know a little about chemistry, you should realize that this ozone compounds, you know, with a O3 can enter very easily in oxidation reaction with volatile compounds that can be oxidized very, very quickly. So that was, we wanted to look on these volatile compounds emitted by figs. Oops, and so we want to study this ozone effect on the interaction, looking as usual both to the plant and to the pollinator. And we study that first, of course, in Ficus carica with our model system, because it's easy to go, mainly when you cannot travel anymore. And also because in the Mediterranean region, we have a lot of this ozone, you know, concentration. And with a very, for example, this red spot are concentration of ozone of 200 ppb with a quite high concentration of ozone in the atmosphere. So we look at the effect on the plant, Ficus carica, but also on the insect, and see how this chemical interaction can be maintained in regard to this sometimes pollution that we have in our areas. So, oops, we put this fig tree. So we use it, we do two types of experiments, and today I will not be able to show only one. For example, we look also at a gradient of, as you can see in the Mediterranean area, we have a gradient, we have a place that are less polluted than the one. So we look at fig emission in different places in Mediterranean area. But also we do experiment, and I find them a little more fancy, a little more fancy to explain to you. So we put some figs in the greenhouse and in this glass chamber. And so we always have a control. And in this chamber, we put this 200 ppb concentration that I told you we can have sometimes during hot summer in Mediterranean region. So we put this fig tree for 3 hours of this 200 ppb ozone concentration, and we extract the odor of the fig at that time, and we let them a little longer, 5 hours, and after that we stop everything. And after sometimes we look at also at the odor of this fig after ozone exposure. And then we analyze them, as I showed to you already, on the gas chromatography and mass spectrometry analysis using thermodesorption techniques. So here you can see the result you have using a redundancy analysis. It's a type of PC analysis where we compare the relative proportion of the emission of these volatile compounds regarding of the treatment and the time of the treatment. And you can see here in green, you have the control, so very well separated from the other one. And here you have the odors of the figs, where you have this 3 and 5 hours of ozone concentration. And here is where we stop it, and we have another cloud of these individuals that are closer from the control. Means that after O3 exposure, it looks like the fig tree can also recover the normal emission. So, but on this figure, you can clearly see that ozone exposure affects the emission of VOX by the plant, and due to the degradation of the VOX in the chamber. So to analyze this degradation of the VOX in the chamber, we add a reactor where you know, it's only the volatile compounds emitted by only the volatile compounds emitted by the fig. So we, you know, they are pushing in this reactor, and it's only here that we had the ozone, the O3 or ozone, and we can have the reaction of only the volatile compound to ozone. And we use a different concentration. We use from 40 to 200 ppb of ozone. And here, as a result, same analysis as before. It's a relative proportion of the attractive box this time, and in regard to the treatment of a different concentration of ozone. So you can see that, you know, even at 40 ppb, all the compounds emitted by fig really change. And you have, it's very different from the beginning. And of course, when you have 200 ppb, it's even more different than when you have only 40 ppb. So the exposure of ozone concentration really affects the relative proportion of the attractive box for the pollinator in the atmosphere. So as we always do, now we know what this ozone concentration can do on the emission of figs. So now we look at what the answer of the insect. So we use always this white tube tests that I present to you. And just to remember you, when you present to this fig wasp, you know, volatile emitted by figs, they are really attracted to the fig. But when you present to this fig wasp some the same compounds, but after having been treated by 40, 80, 120, or 200 ppb of ozone, you can see that these compounds are no more attractive, and the pollinator can no more recognize the volatile compounds. So it's changed in the volatile compounds induced by the ozone concentration has, of course, an effect on the fig wasp attraction, even at low concentration. So the VOX emitted by fig tree are really affected by this emission of ozone, and also so we make some other experiments showing that it's a reaction of oxidation, and this reaction of oxidation really affects the pollinator attraction. So I will finish very briefly because I'm sure I am a little long as usual when I speak about figs, but just to show you very quickly that also as an evolutionary ecologist, we make some, not experiment, but some observation where we show that chemical ecology is also very important for speciation. And we study that in the Philippines on a ficus species, Ficus septica, in this very fragmented landscape of the Philippines with a lot of small islands. And you can imagine that for pollinating wasps, it will be difficult to fly from this place to this place, or even more to Luzon, a little north of the Philippines. So what we have done, we have looked at the genetics of the fig tree and the genetic of the wasp. And if you do the right analysis on genetics diversification, you can see that you know, the spatial variation of the genetic of the fig tree and of the genetic of the wasp is really are really confounded together, and you have a group of this Ficus septica in Thailand, a subgroup in these two islands of Luzon and Visayas, and a third group in Mindanao. So it's really a nice match between the genetic of the spatial variation of the genetic of the fig tree and the spatial variation of the genetic of the fig wasp. But if you add the volatile compounds, you can see here that, you know, in Taiwan it's okay, you have exactly the same geographic variation, and in Mindanao also. But in these islands that, of course, are different small islands together, the odor from the North Islands is quite different from the odor from the South Island. So it's a spatial variation of this odor. And if you look now at the wasps, you can see a tendency of the wasp also to change, and particularly to change in color. So it looks like we are just on a process of speciation, and that are already marked by change in odor of these receptive figs that evolve more rapidly than the speciation. And we hope that if somebody will do the same study like in 20 or 30 years, and I'm sure that we will have a new species of wasp with developing in this area. So chemical ecology can also lead to understand more easily how speciation occur in this very tight interaction between a fig and a fig wasp. So, and we have a speciation, of course, that will be driven by the divergence in this fluorescent. So through the different phase of plant reproduction, as a summary, I can say that, you know, these chemical signals are responsible for this partner encounter in space and in time, that they determine pollinator specificity. I will not have time today to explain to you, but they are also important because you have some parasite on this fig and fig wasp system, and this parasite and anquiline also use the odor that are elaborated by fig, emitted by fig during the development. And also, I will have time to show to you also, but these odor are very important to attract seed dispersor. So shaped by diverse but integrated selective pressure, chemical signal in this system really appear to be the lord of this whole elaborate dance that you have around the fig tree, that are very often called the queen of the forest. And but this, you know, lead to another question: what is the lord of the chemical? How this chemical, you know, can change so rapidly? How you know, the insect can achieve this fine tuning? So now we are starting a study on the genes encoding for biological active scent compound and the frequency of mutation, or how they are regulated, you know, to induce this very rapid change in odor composition. And of course, we look also on the olfactory perception in insect, and what are these genes that would allow insect to learn about fig odors. So I'm done with this a little too long presentation, and I will want to thank, you know, all the many students and all my colleagues now, Magali, Candice, and Saoli, and for all this study. And now I am ready for questions. And I want to thank you for listening.
Martine?
Yep, I'll try.
Yeah. I must say, this was a wonderful presentation. It opens up a new world, which at least I suspected it existed, but really didn't have the large scope you introduced into my mind, and it really gives a wonderful overview, and the complexity that we are studying in ecology, and I think that makes the system study, well, interesting, challenging, and very, very difficult as well, because we are entering complex systems, really complex systems. So we agreed on that. Victoria would take on the questions, and there is someone who should be reading the questions from the YouTube and Facebook. So let's hear about that. For myself, I have a question for you, which then I leave to Victoria. So I'm just preparing her, so she's not so surprised. And as you know, I'm coming to this from a different point of view, but I'm looking at insects that may cause diseases in humans, and that may lay eggs in specific plants. Would you say, and this is far off your model, but would you believe that, for example, Aedes aegypti would have a chemical sensor for the plants where they put their eggs? Martine?
Parece que tiene un problema con el ancho de banda, dice.
Ah. Entonces, mientras esperamos a Martine, qué suerte que vino ahora porque pudo terminar la presentación justo.
Sí.
Digamos, no quiero, digamos, encontraste hay muchas preguntas.
Hay una pregunta en el chat nuestro de Florencia que dice cuáles son las perspectivas a futuro teniendo en cuenta la tasa de polución.
Claro.
Yo también tenía preguntas acerca de cuándo ellos decidían por la maduración del fruto del ficus, en qué momento, o si lo medían a través de toda la maduración y medían esos volátiles, o cómo tomaban la decisión de ver cuándo medirlos.
Martine? Nope. A ver. A ver, ahí se desconecta, por ahí se reconecta.
Sí, quizás. Yo estoy viendo a ver si me manda mail porque a veces me...
Sí, le envié un mensaje para ver si...
Sí, yo también pensaba en esto que dice Flor de cómo el cambio climático, o sea, en la atmósfera, cómo va cambiando, eso de la polución o los cambios, cómo influyen en sus mediciones también.
Sí,
Si hicieron experimentos como a lo largo del tiempo en las mismas zonas y vieron algún cambio o alguna sugerencia que se pudiera, como ella también fue a la COP y eso.
claro, eso...
Luca puso en YouTube que enseguida volvemos con las preguntas, así que